Thanks for the kind invitation. What I'm going to talk about is the topic of research in my lab since about 10 years. We worked on the idea of the possible existence of progenitor or stem cells in the kidney just to address what you know is a relevant clinical problem, as Dr. Herrera has already beautifully shown before. Indeed, I'm a clinician and I continuously face situations like the one that is shown in this picture. These are the cases of two kids. Both had a sudden onset of nephrotic syndrome in the first 2 years of age. Both were resistant to steroid treatment. Both had a bioptic diagnosis of FSGS with a negative immunofluorescence. Both were treated with calcineurin inhibitors and were resistant also to the immunosuppressive treatments. We did exome sequencing, and there was no known genetic alteration in both the kids. But then, over the following 2 years, the two kids had a completely different outcome of the disorder. On the picture below, you can see the evolution in a second biopsy that both the kids had. One of the two kids had a persistent nephrotic proteinuria that remained at about 20-30 of protein-creatinine ratio and evolved toward a pre-end-stage kidney disease with the creatinine that is currently around 3 milligrams per deciliter. And the other kid had a spontaneous remission of the disorder, was treated only with ACE inhibitors, and underwent a complete disappearance of proteinuria. He has currently a very good biopsy, even if the picture at the onset was extremely bad, and a normal creatinine function. I can tell you that this remission persists now that more than 4 years have passed. So, which is the reason why two kids with apparently the same disorder, of course, there are many things we still don't know in nephrology, so it may be that these are not the same disorder. But currently, we define both of them as focal segmental glomerulosclerosis, steroid-resistant nephrotic syndrome, not related to a genetic cause. There may be, of course, many reasons why they had this different outcome. This may indeed be two unknown different disorder. It may be that the two kids had a different genetic background, but the experience of a different outcome of similar disorder is something that people in all the organs make every day. For example, even when you make a wound on a skin, you can have a completely different outcome of the wound, depending not only from the severity of the injury, but also on the individual capacity of each subject to repair the injury. And we know that all this kind of glomerular disorder that we define under the pathologic definition of focal segmental glomerulosclerosis and that are characterized by an important proteinuria are related to injury of a very important cells of the glomerulus, that is the podocyte. The podocyte is the main cell that is the constituent of the glomerular filtration barrier. It's a very particular cell. It's very similar to a neuron, highly specialized, post-mitotic. It has lost the capacity to divide, just because it is such a complex cell that makes such a difficult job, keep the glomerular filtration barrier together. And so the possibility of its division may be a danger for the structure of the glomerular filtration barrier. So the problem is that when this cell is lost, we have proteinuria, and there is a direct proportion among the entity of the podocyte lost and the amount of proteinuria. And then the problem is if these cells can be replaced. And still, we didn't know that until very recently. Very recently, there have been several studies that have proposed the possibility that there could be some regeneration of podocytes after glomerular disorder. But this has been a very controversial topic and very difficult to address, because for sure the number of podocytes that can be found after a glomerular disorder is usually reduced, and it's very difficult to establish even if it's increased again, because you should make repeated biopsies of the patients over time. And so the only way to really see if these cells has a capacity of replacement is to use transgenic model and lineage tracing techniques. With these models, you can irreversibly tag a specific cell, in this case a podocyte, in a way that these cells becomes of a color, for example, green, and only the progeny of these cells will be of that color, and whatever other cells that come from another source of the kidney will be of a different color. In this particular case, we use a transgenic mouse model where the podocytes are tagged in green upon treatment with tamoxifen. In this manner, you can decide when the podocytes will become green. All the other cells of the kidney are red in this system. That is a podocin I3 estrogen receptor transgenic mouse coupled with a tomato GFP reporter. And so you can beautifully use this model just to know which are the podocytes that were generated before the injury and which are the ones that were generated after the injury. Because when you remove tamoxifen, all the newly generated podocytes will be red. So the old podocyte before the injury will be green, or eventually their progeny. The newly generated podocytes, if they come from a different source after the injury, will be red. Okay, so what we did, we took mice, we induced a model of focal segmental glomerulosclerosis by using adriamycin injection. This is a widely used model of focal segmental glomerulosclerosis. And we went to see what happened to the mice 1 month after the injury. We did this because we thought this is exactly what happens with patients. They arrive and you don't know which of these patients will go well and which one will go bad. You will know it only after. But with these mice, you can look 1 month after what has happened in the mice that had the remission of the disease in comparison to those that instead progressed. And look if there is a different number of podocytes and if there were new podocytes generated. That's what we did. We divided the mice into different groups. Those that after 1 month had undergone remission of proteinuria. This is something that commonly happens with this model. It has an heterogeneous outcome, exactly like it happens in patients. And some other mice had a persistent proteinuria that remained over time and brought to an abnormal kidney function. And what we saw was that in the mice that had undergone proteinuria remission, there were some new podocytes that were red. We could establish it by making a labeling in blue with synaptopodin. And so we could see that there were some new podocytes. Well, if we could turn off the light, probably you could see it a bit better. Well, and there were some podocytes here that were red and blue. So this means that these were generated after the injury and were coming from the source that were not the pre-existing podocyte. Otherwise, they would have been green. We counted these cells, and we saw that in the mice that had undergone remission of the proteinuria, the new podocytes had replaced about 30% of the lost podocytes. This model allows you to beautifully count in extremely precise manner all the podocytes. And those that had persistent proteinuria basically had no regeneration of lost podocytes at all. So this model tells you that there can be podocyte regeneration after injury and that this regeneration occurs from an external source, somehow not from the pre-existing podocytes. Otherwise, they would have been green. But the problem is, where do these new podocytes come from? Okay, so that's exactly the topic of my work, as I told you, since 10 years. And over the years, we have been establishing together with other groups that there is a niche of progenitor within the Bowman capsule, and there are also other progenitors scattered along the nephron, and the distribution of these cells is extremely similar to the one that is observed and then has been recently described also in the fish and in the insect, and is highly conserved across evolution. what is usually, it doesn't go this way, but the first species where this was first, this system was first characterized was human. And here it's very easy to identify these cells because they are all characterized by the expression of CD133. And in this manner, you can see that there are these progenitor cells that localize at the urinary pole of the Bowman capsule. These cells, if you recover them, have the capacity to generate both podocytes as well as tubular epithelial cells. Then there are cells that are distributed along the parietal epithelium of the Bowman capsule and that somehow already have a commitment toward the podocyte phenotype. So they have a mixed phenotype, express CD133, but already start to express podocyte markers. These are those cells that were commonly known as parietal epithelial cells. And then there are some cells scattered along the nephron and distributed in specific compartment of the tubular segments that are tubular progenitors. The problem is that while these cells can be beautifully identified in human, the CD133 is a species-specific marker. This is this way in several organs of the human body. And so you cannot use it in the mouse to detect exactly the same population. Of course, in human you cannot do transgenic models, so then the result is how to study exactly the same population in the mouse. And that's a problem that we have tried to solve in the last 4 years. And we had the idea to use a marker that is PAX2 that is always co-expressed with CD133 in the human renal progenitors. So that's the reason why we established a new model, a transgenic inducible model for lineage tracing that uses PAX2 as a promoter. Then you can give doxycycline and activate the expression of whatever reporter under the control of this promoter. And when doxycycline is removed, only the progeny of the population that was labeled will be tagged. Okay, so in this manner, you can rule out the differentiation, because it's a closed system. We use different reporters. First of all, I will show you the picture taken in this mouse with the confetti reporter. The confetti reporter is a reporter that allows the stochastic expression of 4 fluorochromes under the control of the promoter that you have chosen. So whatever cell express PAX2 will be red, blue, yellow, or green. And if we turn on the mouse with doxycycline at the 5th week of age, so when the animal is already adult, we see that there are some cells that become colored in a stochastic manner, and these are some cells around the tubule, as well as cells of the Bowman capsule. If we do it at the 2nd week of age of the animal instead, we see exactly the same picture, and then we can remove doxycycline and follow what happens to the cell up to the 5th week of age. And we can see that some of the cells of the capsule move inside the glomerulus and become podocyte. Here you can see a detail. So that's how the PAX2 labeling appears at the 2 week of age of the animal, and that's how it becomes if doxycycline is removed, and then these cells are followed up to the 5th week of age of the animal. So means that during the growth of the glomeruli, some cells of the capsule are recruited to become podocytes. And they contribute to glomerular growth. Now let's see what happens if instead the system is directly turned on at the 5th week of age of the animal. So when the growth of the glom has already ended. And here you can see that PAX2 is only expressed, so renal progenitors are only localized in the capsule, and then as scattered cells along the tubule, exactly with the same distribution described for CD133 in human. If we remove doxycycline and follow up what happens to the parietal epithelial progenitors up to the 9 months of age of the mouse, so up to when the mouse is old, we see that there is no new recruitment of podocytes from the Bowman capsule. So this means that the parietal epithelium is progenitor for podocyte during the growth of the kidney, but then doesn't generate new podocyte in a turnover manner during the life of the animal. Now let's see what happens after injury. Because in many organs, you know that progenitor or stem cell system are active after injury, even if there is no turnover during the life of the animal. Okay, to try to establish if these cells had a role in the generation of new podocytes that we had seen at the beginning, we used another reporter, that is the tomato GFP reporter. Here you can see the control of the mice when there is no turn on with doxycycline, so the kidney is only red. When you give doxycycline to the animal at the 5th week of age with the tomato GFP reporter, the progenitor will be green. And you can see them in the Bowman capsule and as scattered tubular structures. Here I show you in detail the picture of the Bowman capsule where the progenitors are green. And there is nothing inside the tuft, as you can see. Okay, now we have done exactly the same experiment that I have shown you where the podocytes were labeled. We have divided the mice into groups, and we have looked what had gone on in the gloms of those mice that had a persistent proteinuria 1 month after injury in comparison to those that had remission of the disease. And we could see that in mice that had persistent proteinuria, there were no cells, no new green cells inside the tufts of the glom. The cells were only in the capsule, or sometimes were generating some hyperplastic intraglomerular lesion, but without any shape. While in those mice where there was proteinuria remission, we could see some green new cells perfectly integrated within the tuft. And here you can see the green cells that had food processes, which is a specific characteristic of podocytes, and that also express synaptopodin in blue. So these new cells coming from the Bowman capsule were podocytes. Here you can see again a picture of the food processes, and here the detail of a glom. Here the progenitor labeled in green in the capsule, a clear new podocyte that has been regenerated within the tuft of the animal, the perfect structure of these cells that has clearly primary and secondary food processes. Now, you can count the new podocyte generated by these progenitors, and you can see that basically you get exactly the same data that I showed you with the first model. Newly generated podocytes within the tuft of mice with persistent proteinuria, only lesion, hyperplastic glomerular lesion, in the glom of the mice with persistent proteinuria. So the process occurs, and it's determinant to establish if you will get the remission of the disease, because it leads to regeneration of about 30–40% of the lost podocytes. And this makes the difference. But then the point is, if we're going to go to treatment, can we modulate the process? So we have made a screening of a library for different compounds that could upregulate the differentiation of these progenitors. Because as you have seen, the fact that you get remission or progression depends from if these cells are able to acquire a full differentiated phenotype of podocytes. And in this manner, we selected a compound that is an inhibitor of GSK3 that can enhance the differentiation of progenitors into podocyte by increasing their response to retinoic acid. That is an endogenous differentiation factor for these cells. So it enhances their capacity already in vitro to become fully differentiated podocytes. We gave this molecule to the mice, and we could see that in the mice treated with the molecule, there was a substantial increase of portion of the glomerulus where new podocytes were generated and regenerated. And also the number of mice that underwent remission significantly increased. Here you can see a detail of regenerated gloma. The part in green are all those consisting of new podocytes generated by the progenitor of the Bowman capsule. And here you can see that the molecule induces remission of proteinuria and also a considerable improvement of kidney function. So this means that progenitor-mediated podocyte regeneration and glomerular healing can be enhanced with drugs. So here you have seen that glomerular progenitors can really represent targets for drugs, and I think this is a completely new strategy for thinking to new treatment for glomerular disorder. But they are also an excellent tool, especially for modeling of disorders. Here you can, this is a paper that is currently in press in JASN. We have established the method to purify and amplify these cells from the urine of patients with glomerular disorder. These cells are extremely rare to detect, but somehow they can be amplified. Even if you get a single cell in the right condition, they can be amplified to give several million of cells, and then you get a culture that is a personalized modeling of the disease of that patient. These cells are progenitors. We have demonstrated this by injecting them into SCID mice with glomerular injury, and we get differentiation into podocytes in vivo. In vitro, the cells can be exposed to differentiation media, and they acquired RNA and protein and also electron microscopy structure of podocytes. And then we use them to support diagnosis of genetic kidney disorder in those kids where, after exome sequencing, we have, for example, mutation. We find mutation that we suspect may be the cause of the disease, but since they are unknown, always and frequently missense mutation, we cannot be sure that they are the cause. We get the culture made from that patient, and then we have a perfect modeling to functionally study the mutation in the podocyte of the kid, because these progenitors can be differentiated into podocyte in culture. We have already done this. Here, these are 3 patients. For example, 2 had the podocin genetic mutation. One was an compound heterozygosis. The other was an homozygosis. And the third one had the mutation in LMX1B, was unknown, and we suspected could be the cause of the disease, but we could not be sure. And you can clearly see that the podocytes obtained by in these kids have extreme cytoskeletal abnormalities. They can be studied. They are less vital. They are prone to die and to detach in comparison to those of patients without genetic mutation. And so we think that this may be a fantastic model to study the disease of the patient. Also, for example, to study the epigenetic contribution of the pathogenesis of a disease in a single patient. And we propose a strategy where we can make exome sequencing and then amplify the culture of these progenitors from the patient, and then use the culture to functionally study the mutation, or, for example, for drug screening. So as you can see, this population of cell is really opening a new era, I think, a new eve in kidney diseases study, and we will have a first meeting on renal progenitors in Florence next year. So you're all invited to participate. And this is the group of my people that did all the work. Particularly, this work was made by Laura Lasagni. And I thank you for your attention.